Good day everyone. We are here to present Microscopy 1, Sample Specimen Under the Microscope. I am Stephanie Nicole Binarido, together with my group mates, Aguilar Marian, Carcer Esther Joy, Rebedin Sharilyn, and Santo Domingo Melanie. Microscope is a crucial tool in various scientific fields because it allows us to visualize and observe objects and find details of plants and animal cells that are too small to be seen with the naked eye. With this, we are to observe some plant specimens such as monocot, stem, dicot stem, moss, fern, male cone pollen, and flower lily, to exercise our skill on using microscope by observing those specimens. Methodology. Here are the steps we followed. First, set up the microscope. We check the lenses, clean the slide, adjust the light, and prepare the stage. Second, prepare the specimen slide. We used ready-made slides with the different plant samples. Third, observation under magnification. Each specimen was viewed under scanner 4x, low power 10x, and high power objective 40x. Documentations. We record what we saw and compare the details to each magnification. To our result and discussion.
First, the Pinus male cone with pollen, a gymnosperm. Pinus male colon with its small dot-like structures show the reproductive organs of the pine tree. At the higher magnification, we can see the microphorophyls and pollen grains. Next is fern protalium with mature sporophyte. Pteridophytes. The fern protalium is the heart-shaped structure of fern that eventually grows into a mature sporophyte. We observe its green cells and reproductive organs clearly.
Moss protonema or mosaine mosses. Mosses protema is an early stage of moss development. Under the microscope, we saw a delicate threaded-like structure that formed the moss plant.
Lily ovary cross-section. The lily ovary contains ovules, which are crucial for reproduction. At high magnification, we can clearly see the internal structures, including the female gametophyte and ovule. Elodea stem section. Under scanner, small rounded shapes with some clarity, while for low power objective, the outer edge is visible filled with small shapes. Under high power objective, detailed view of tiny, rounded structures are packed together. This Elodea stem, a monocot, reveals closely packed cells. This aquatic plant stem is adapted for water absorption and transport. For Dicotyvia young root, the Vicia bean root cross-section shows the structure of dicot roots. At high magnification, we can observe the root's vascular tissue and other detailed structures.
For adding more explanation or information, we saw two rounded shapes that show the basic form of the dicot root. As we focus the microscope, the outline of the root became clearer and small inner structures started to appear. When we adjust the focus more, the center of the root looked compact and detailed, and the tiny cell could be seen more clearly.
In conclusion, through this laboratory activity, we had the opportunity to observe various plant specimens under the microscope, which enhanced our understanding of their microscopic structure. By adjusting the magnification and focusing on different parts, we were able to identify essential cellular details, which will aid us in our further studies of plant biology.
Furthermore, in this activity, we learned how properly use the compound microscope and how important it is studying very small structure of plants. By observing different specimens, we saw how each magnification helped us notice more details that we cannot see with the naked eye. This made us understand the difference between plant parts such as stem, roots, spores, and productive structures. This activity also helped us practice focusing microscope, adjusting the light, and handling the slide correctly. We become more familiar with how plant tissues are arranged and how their shape change depending on the specimen. Overall, the lab activity improved our observation skills, our patience, and our understanding of plant biology. Thank you very much. During the activity, our group worked together in preparing the microscope, checking the slides, and observing each specimen. We took turns adjusting the focus, changing the objectives, and describing what we saw. We also recorded our observation by writing notes and taking photos of the slides when it possible. We documented the appearance of each specimen at different focus levels, starting with its overall shapes and then its smaller details. We also compared what we saw with reference image to help us identify the parts correctly. Our documentation show how the specimen changed in appearance as the magnification increased, and how different plant structure can be observed clearly using the microscope. This helped us better understand the features of each samples and improve our skills in using laboratory equipment. Thank you everyone and God bless.